So it gives me great pleasure to introduce Dr. John Medaglia today to give our topics in rehabilitation science lecture series for this month. So a little bit about Dr. Medaglia. Many of you may have know him or have heard of him before. So Dr. Medaglia received his undergraduate degree from Drexel University before completing a PhD in clinical psychology from the Pennsylvania State University in 2014. He subsequently completed a joint postdoctoral fellowship within the Jefferson Moss Rehab Research Institute and the Penn P32 postdoctoral training program, which many of you are familiar with. During his time in the program, he received guidance from a variety of mentors across the two institutions, and in 2015, he received the prestigious National Institutes of Health Directors Early Independence Award to study brain stimulation-based cognitive control treatments in health and stroke. Dr. Medaglia now serves as an associate professor in the Department of Psychological and Brain Sciences at Drexel University, where he directs the Cognitive Neuroengineering and Wellbeing, or CogNEW laboratory. His laboratory applies models and methods developed in neuropsychology, cognitive neuroscience, and network science to optimize brain function in healthy and clinical samples with a focus on how people control their behavior and how control theory can guide non-invasive brain interventions. The laboratory uses network-based approaches in neuroimaging like MRI and FNIRS, brain stimulation like TMS and TDCS, and behavioral assessment to meet these goals. Lab members additionally study neuroethical decision-making. Dr. Medaglia received support from the NIH and donors to support his research, and under his mentorship, many of his students have received independent funding to support their projects. So I will leave it to Dr. Medaglia now to take it away with this talk. Thank you.
Sure. Well, thanks for that generous introduction, and it's always a pleasure and honor to be back around here with some familiar faces and some new faces. I rewrote a bit of what we've been doing recently for this audience, really thinking back to my time as a T32 fellow at Moss Rehab, and the decade or so since then, spending a lot of time with neuroimaging and brain stimulation, and I get sort of reflective at that point when I come back to old places. And what I really wanted to do today was give you where I think the optimistic cases for a lot of what I think I've learned about brain networks. Probably give a fairly strong opinion about an area of research that we just for some reason don't spend a lot of time on. And I think that's kind of a shame. So I'll try to justify that with a couple bits of data. And then in the end, come around critiquing my own agenda completely and sort of pointing out, we got a lot of work to do here. But I think, in those ensuing years, I have a couple slightly more progressive things to say about it where we can make progress. So, first of all, thank you to the Shriers making this series possible. I think it's great when you have folks recognizing the value of institutions in series like this. And again, I'm sincerely honored to be here under that auspice. There is some CME credit on board. And so I think under that model, I did think about, some of the planning committee here. So special thanks to Umi for giving me the direct invite and many friendly faces here supporting me here. No financial disclosures. I want to talk about the strengths and weaknesses of studying structural brain networks in neurorehabilitation. I'm going to use an approach that many of you may have heard of as graph theory. There's a mathematical basis for this. I'm not going to spend a ton of time talking about the details actually, because in the end, that's really just a method for dealing with the problem of brain networks in rehab to me. I want to talk a bit about why would you do this? Why would you use these concepts to study rehab outcomes? With a couple of bits of our own data and some promising NIH-reviewed material we hope to be pursuing in the next 5 years or so. And I want to just talk again about why are we doing so much of this research and why isn't it helping people faster? And not to be too politicized now, but I do think that there's kind of an undercurrent affecting all of academia and pressuring us in a number of ways, but one of which is, well, I go and I spend a lot of this time in NIH dollars doing these things. Is there any sort of due diligence to getting it back to any one person? I personally take that very seriously. It was my hope maybe 15 years ago as I got really deep into neuroimaging, I would have a better answer to that. I'm afraid I don't have a great solution, but I do have, I think, hopefully a new perspective on where we could go next. My lab, again, just downtown there across from Penn at Drexel University. We do three things, broadly speaking. We do what I call cognitive network neuroscience, which is really just cognitive neuroscience, but we think about networks. Precision brain stimulation. So I'm going to talk only a little bit about that at the end of today, but we think a lot about where and when to stimulate people with electromagnetic currents to make them better. And so I had actually on mind an altogether different talk just on that, but I actually decided to step back a bit and talk about some of what I think are prerequisites to that effort. And I like to just tell people this is sort of my favorite secret hobby interest, where every once in a while we publish something on decision science and neuroethics, where we don't so much have opinions about what's neuroethical to do, but we study why people think certain things are neuroethical to do. So we have a little bit of the lab focused on that at any given time. So let me get right into the content that might interest this audience the most, which is when we think about some kind of syndrome. I'm going to talk a lot about stroke today as the disease model. A clinician only has access to so much. And so in the routine standard of care in the United States, you're pretty lucky, first of all, to have access to something like an MRI in a lot of cases. And so you're often in a sort of privileged community just to get that. And the clinician might work with a scan that is kind of pretty easy to obtain, pretty reliable, tells us a lot about a person. And hopefully most of you can see up here, there's a pretty visible, I'll just use my hands like I do in class. There's a big stroke that's very visible. You could have a clinician come in and do a hand-drawn tracing. There's a big effort in trying to automate that to sort of take a lot of that burden and error out potentially. And that's been a lot of anatomical stroke research to date. A lot of classic language models will focus on certainly networks, but they're also going to talk about specific regions and pathways within those networks. And so we have this sort of parts-based habit in cognitive neuroscience, which is, well, we spend decades trying to understand the role of a region and its connection to something else. And even still in this circuit that some of you may recognize as the arcuate connecting Broca's and Wernicke's areas, even the definition of those parts and their roles and specifically what they do, virtually at every level, have some debate associated with them to this day. So that's kind of one aspect of the work, is figuring out what are the parts, what do they do, what are they for? And then we come in and we might lesion that connection, we might lesion one region or the other, and then we try to make some inferences and inform our rehab. When we have something like a hemiparesis, we often have something that has a pretty focal effect on a corticospinal tract descending down through the spine. We trace the pathways. We love wiring diagrams like this because we can kind of say this connects from A to B, and so doing some kind of break might give us some predictable outcome or clinical value. So all of these things are things we have done. They're all things I think we should continue doing. I think one of the things that started to bother me increasingly over time is this is kind of a general statement. It's hard usually to distinguish the exact sources of dysfunction. I think stroke's really interesting because it lets us think about a couple of problems. So I want to sort of, while focusing on stroke, highlight what I think that disease model lets us do scientifically, and I hope clinically. But you can also think about more kind of graded disorders where somebody has a more subtle injury or progressive dimension and think about how these concepts apply. Well, let's talk about stroke. Here's one of the problems. When the lesions are large and have a lot in common, you can't really know what specifically matters. What I mean by that is if you get a lesion overlap map and you know there's this big mass, and a lot of people have it, the problem is about variance. It's hard to know then if all of that is pretty fixed, what varies with that particular infarct. So that's a general pattern we see where we're able to make kind of these broad brain behavior associations between maybe some lesion and outcome. But because we have all of that in common, the problem of variance and individual variability is sort of tricky to figure out. And when lesions are unique, where I would actually argue they're all sort of unique when situated in any given person's brain, but broadly speaking, if it's very unique, we don't predict outcomes very well. And so we have these phenomena such as silent strokes. As many of us age, we're sort of accruing these over the lifespan, and maybe there's no one symptom that really shows up, and there are a lot of thoughts as to when and that doesn't happen, but it's very hard to predict when something that's very small and focal becomes suddenly symptomatic and suddenly debilitating and a big problem for somebody. Okay. So I was doing this for a while. I started in an fMRI lab with Frank Hillary at Penn State researching executive function mostly, and sort of like, why does brain activity go up? And is this compensation or reorganization? There's a lot of interesting sort of philosophical debates attached to the imaging. We spent some time there. I moved on to functional analysis and networks at Penn with Danny Bassett and Sharon Thompson-Stoll and Roy Hamilton. Moved on to my role over at Drexel and was sort of putting a lab together. And I kept having this problem over and over again. And it became sort of a compulsive obsession in a way scientifically, where I was like, well, there's a causal cascade we sort of know a lot about in some of these syndromes. In something like a stroke, you have some kind of premorbid vasculopathy. Typically, there's some risk factor. Sometimes it's hard to pin down. The person might have a brain bleed, and then they lose some structure. Right. And so part of the advantage to us as a scientist is, again, you can see it and you can ask somebody to draw around it and you can debate about it. And you can have, different reliability across different raters, and we can do that work. Lesion size and location are familiar, famous, fully justified variables when you think about that. Yes, how big something is and where it is predicts this outsized share of what happens. But the problem and the obsession for me was every single time I damage anything anywhere, by definition, in a connected system like a brain, it's also damaging a connection, either directly or indirectly. I've lost a region, it had inputs and outputs to it. I lost a path, that was a connection itself. Anytime I perturb this structure, there's an unasked network question somewhere. So the question to me became, well, how can I get at that, and what questions are worth asking in that space? Presumably that leads to some sort of brain dysfunction or not. Is it silent or is it symptomatic? And then possibly there's some functional adaptation broadly defined and some behavior changes, and maybe it'll get better or worse. And so we do this through this pathway, whether or not we're working with all of those elements. And so what I found interesting was when you look at the imaging literature, we have a lot sort of focused on the lesions and the structure, and the sizes and locations. We actually have a lot focused on the function, trying to figure out what activates or what's functionally connected. But we kind of forgot this idea somewhere along the way that the roadways are a mediator and constraint for, well, a very large proportion, presumably, of how brains communicate. There are exceptions we can talk about, but let me just kind of build the case here. In normalcy in a brain, I like these road metaphors because there's a lot going on, even in a pretty simple roadway, right? You have vehicles traveling at high rate and mass. You have lots of little populations of people that look sort of stochastic, but you notice that a lot of the traffic flow is organized in space and time by the underlying roadways. And so you can have some sort of inference from just the fact that there's a road here, there could be traffic at all. And within the context of that roadway, how does the traffic work? These are the kinds of questions people start to ask in structure function analyses. When you lose some kind of pathway, you try to reduce the path structure and capacity. You do it intentionally or not. Well, obviously, something has to happen with the rest of the traffic. It's going to change. The hard part about that, and I think the story I'll tell today about networks, is it's not really straightforward to know what you should expect from that, even from the structure, right? It still depends a lot on the particular pathways, their configuration, how they were used in that particular point. But if you don't encode that pathway into your model of what is broken and how it changes and how to fix it, my concern is you're never really going to figure out how that traffic should be readapted at all. And so that's where we're going. So can the newer imaging help us with old problems? I'm a theoretician in disguise, is what I like to say recently. What I'm really after when I'm talking about this is a theory about how the brain structure organizes the function and the behavior. I want to know what dynamically does it. I want to know what the brain computes within that context. But I want to know when it changes and why, and who's going to get better or not, and can I do something with that? So in the standard clinical care, we'll get an image like this, we might see the stroke. And the first thing you might notice, and many of you are familiar with this, is you might have this sort of obvious lesion mass and the locus of that infarct and its breadth and its geometry, the things you can see and you can draw. You might have these more distant effects. So you notice here, this is more to a sort of lateral parietotemporal lesion is maybe the first thing your eyes go to. And then down there, you might notice down of the sort of inferior frontal gyrus, these gyri are sort of thinned out. There's maybe been some atrophy, diaschesis of function, other things like this. So you also have this problem, even in the T1 scan, of like, what was the initial event and what is the other stuff? And what matters about that? What is a change immediately? What is a change later? What does it do for recovery? Underneath of that, in a healthy person, we've had now for the better part of 20 years, diffusion tractography methods that allow us to make some guess about the white matter pathways about it. So we'll talk a bit about the pros and cons of that, but the technique's been around a while and in some ways has gotten a lot better. And so now, even before I worry about what's going on down here in the stroke, I can ask a lot about the structure of the roadways and how might they have something to do with the function. And indeed, there's a whole line of studies on this now for the past 10 or 15 years. Immediately, when you start to see a stroke through this lens, or I have as I start to look at these brains case by case, is I never see a focal lesion in my model for how that works. I see a connection issue. I see a loss of regions and connections that not only have this particular lesion profile, but as we'll see throughout the next few slides, lots of indirect consequences we can get directly out of imaging the structure. And so to me, I have to worry about reliability first. So if you're a bold researcher, you're probably famously abused by the fact that you can't get this underneath this fundamental hemodynamic coupling phenomenon, and the scanning is only so reliable up to a point. And so I don't envy my fellow MRI researchers in the sense that like the technology could be a lot better. And I think that's no one's fault. So you aim the same kind of question at the white matter pathway question. What you really love to see, what I would love to see, is you can go back to even very old textbooks like Henry Gray's and start to look for known features like cortical spinal tracts. And we should expect probably everybody has that in this room. Maybe there's some individual variation. Sometimes genetically it doesn't happen for you, but we have a pretty good shot that any given brain is going to have this. So what people started doing in the late 2010s and a little bit thereafter was start to build tractography algorithms and try to find these things. And so you would see something like this in the middle, a little bit over a decade ago, where you can kind of visualize there the blue, which is the descending major part of the corticospinal tract. But what you're missing are all of the radiations, right? You would really like to see it fanning out within the gyrus to innervate and put outputs through that pathway. And so we want to see everything around those red lines get some content. So some clever people modified the algorithms and found, well, if we kind of give a little bit of flexibility and probability to how the parameters go, very quickly, just a few years later, people were figuring out how to use these more probabilistic techniques. They improved the sort of deterministic techniques. And now we start to see more and more maps like this where they're not perfect, as we'll see in the slide, but they do a pretty good job sampling these things that we want to talk about. Okay. There was this justifiably well-cited and famous study by Meyer-Hein, now getting close to a decade ago. But at the time, they came in and they started to say, well, here's the broader problem. Sure, you can find me corticospinal tracts, but it's not a coincidence people started there. This is this big, robust pathway. It descends. It's got a very thick bundle, right? So if you're looking for the best case scenario of a thing to find with this imaging, that's probably what you would find. So they started picking out other structures we're familiar with. They started taking ground truth bundles that they designed into the software and then started asking, well, if it looks a lot like a superior longitudinal fasciculus, if it looks like an arcuate, if it's a little uncinate fasciculus, how small and detailed and tortuous can I get before I do a bad job finding it? Okay. So what the study showed, broadly speaking, and if you're really interested, go read it. There's quite a bit of content there. Reassuringly, these methods had, even at that time, gotten pretty darn good at picking out the structures, even when they involve a lot of convolutions, even when they're very tortuous, even when they're very short. However, as you keep pushing into those dimensions, really short, really tortuous, etc., you start to get a lot of false positives, right? So you got a lot of stuff that like is somewhere nearby the tract you're looking for, but there's like a lot of stuff that really isn't there. And they know that because they have the ground truth in the simulation. So this study got me interested in a couple of things. One, okay, this isn't the worst. We're actually doing a pretty good job getting it when it's there. We have to worry about this false positive issue. People build algorithms for that at this point, so you can kind of trim things down. In addition, what can simulations tell us that we didn't otherwise know? Here, this is the ground truth problem. You're not post-morteming most brains, so it's really hard to know if it's valid or not. We have atlases now, some of which have been around for quite some time, about 20 years, telling us priors on what tracks we should find. So the Johns Hopkins Atlas, there's an ICBM Atlas populated by a number of persons. And reassuringly, in rare cases, people have gone through and meticulously done postmortem studies to show, yes, in fact, when I pull that track structure out from the imaging and then look for it later, we tend to get a pretty good result with some of these. Okay, so what am I trying to say? It's not perfect, but I personally gradually over the years started to say, you know what, like we can probably do a decent guess at this point about these tracks. So let's think about what we could do with it. Back to networks. This is my other obsession. Are neurological disorders always network disorders? I like to sort of pose impossible philosophical questions where the answer is like kind of yes, depending on what you mean, but then also not, depending on what else you mean. So let me kind of talk through that. In the case of aphasia, if you start with something like a circuit model, and it's Broca's and Wernicke's and their pathway connecting them, well, already that's sort of a network, right? It's a single circuit network to many of you. It's a region and a connection, but it's kind of the prototypical simplest possible network. It's a basic unit for what network scientists call networks. However, like the image earlier in the diffusion imaging, it's not just that that's going with the region loss. We have the diffusion imaging scan. So the thing that you can do with a lot of network methods is start to ask, well, if I start breaking elements out, well, first of all, I can measure a lot of stuff about how that was organized before I broke it, and I can measure what changes about it. And what you might immediately see in this toy model is some things will tend to group together. Those are the like nodes colored because they have a lot of interconnections among them. And then there are other things that are a little more disparate. And so this is a concept people usually term modularity. Almost all network systems have some degree of it. Brains, very famously, a lot of studies have shown, are kind of in a weird sweet spot. They're more or less, most of you in the room will be in some sweet spot, sort of balancing how clustered and distant the connections are from one another and how clustered they are with themselves. And so this old idea is the modularity of mind, right? So people subscribe to some extent this idea that there's going to be local specialized functions grouped together, and then they have a sort of minimum path to get from thing to thing. Okay, so that's a lot of network gobbledygook. How do we turn it into something for stroke? Well, if we take this brain and I start to do my lesion masking and overlay, the first thing we start to notice when we do this case by case is even lesions that look really similar have a lot of different intersections with the white matter paths. I'll show us a few examples. In this particular case, we're already expecting, some interhemispheric disruption through the corpus callosum, depending on how forward or back we are in the brain, anterior, posterior. We might have intersected with part of a cortical spinal tract. So already we can populate guesses about what's disconnected from what based on the lesion mass. Once we've done that and we have the render here, well, the network method basically just adds the ability to draw a picture of that. We take a little math diagram called a graph, and we start to look for disconnections. So here's a real case example. This is one of the first ones that we worked with, where we have their lesion mask. That's roughly what's erupted and damaged in their connectome. And then this is the total edge loss in that stroke. So what we started to see time and time again was, even if the lesions are quite small and look pretty focal, and you might have kind of thought of that as like, well, that's like an arcuate sort of lesion. Well, there's all these other possibilities that the other paths are affected too. And we usually have this much broader loss than it might even appear on the scan. Very often it's much larger. Not always, but very often. So those happening so alarmingly that we started to step back and say, well, what should we ask first about that? So we had a couple of ideas. Well, if you look across a bunch of scans, you have the conventional problem of the size and the location. That's familiar and predictive. However, when I kind of say the uniqueness problem is, no matter who that person was, there's kind of, to me, it's hard to say there's the same lesion in any two people, because whatever state that person was in was a different scenario than what it would have been in anyone else. So the next thing that drove me nuts was, how often do I get premorbid data on these people? It's very rare that you get specifically this kind of imaging, much less MNI imaging. Now, there's larger samples data sets. We're starting to talk to other collaborators with these big samples, that they exist, but not frequently enough for us to really get a sense of like, what's the variability out there? And if I don't know the variability, what am I going to predict with? So we were thinking about that problem, looking at all of these scans. And we came up with a scheme like this. Well, we started to say, can I make a brain that would have been what this brain looked like before it was lesions? We started simulating. We started taking out of sample estimates from matched people, matched healthy controls, and saying, well, taking this topology, this network from this person's brain, what would happen if I estimated what it would look like in many other people's brains? Why would you do this? Well, one, it gives you that premorbid estimate, because if stroke is about change, I want to have a guess at that. Secondarily, it also gets me something about individual variability and what I would expect, right? So here's the other problem, kind of a dirty secret in imaging. Not so secret to many, but I kind of think of it that way. Well, we're lucky if we get 40 high quality scans in some of these samples. And so many of you may be familiar with this critique of like, we're just always underpowered and doing too few studies. So I thought about, what's a way we can do this without cheating? What's a way, given all of these healthy scans, and now we have lots of corpora for this, what if I take this smaller sample of strokes, and what if I start kind of case matching them in various ways and then testing how it varies? So what we did was we took from a sample with Peter Turkeltaub at Georgetown University, 39 people. We had sort of curated down to confirm we had high quality both imaging and Western Aphasia battery scores, a global outcome for language performance. We took about 36 controls that were closest matched to that particular subsample, also at high quality, and we multiply them by one another. So we turned this smaller sample of less than 80 people into about 1,400 cases by taking every lesion and simulating it in every control subject. So now what we can do is ask, well, given this lesion, what's the expected average effect in its variability across everybody in the room, for instance? That allows us to say, well, even though I only had one stroke scan, what's the prior I would have on the expected effect across the sample? Working under this at the time, it was sort of a project we were inventing as we were understanding the question. I was starting to come to realize that it might be possible that the simulated estimate does better than the scan. So that's my teaser trailer for what happens next. So the scheme looks something like this. You do it in a healthy person. This here is just a matrix representing all the possible connections when we parcellate the brain. There's a whole nother talk in how we can talk about it and its problems and challenges. We get some network estimates. We do the same thing down here, but what we've done is we've taken some control information to impute the tissue, get a good estimate for the network. Then once we've tested the damaged network, we simulate that in the healthy subjects. And so the scheme now we've replicated a couple times in an ongoing study, and it's working pretty well now. So do the structural networks do anything more than the lesion? Once you regress out the lesion size and the location broadly, short answer is yes, actually about 20% more. So if you're going to do something on the order of 25 to 40% variance in prediction just on the clinical scan, you get actually quite a bit more bang for your buck just taking this measurement out from the scanner in the first place. So the reason we know that is we simulated this large 1,400 person sample. We had their Western aphasia battery scores and a few others. And then we took several popular network measurements. Now, again, I'm secretly a theoretician. I don't know what the hell any of these have to do with cognitive function and outcomes. However, I know something about what they say about networks in general. That's what the network science contributes. So when the network scientist looks through this lens, they're sort of saying, well, what's kind of a corollary of an overall lesion volume? Well, that's just like how many connections are there. So we get to an edge weight measure, okay? Well, it might have something to do with how well or not these big major modules talk to one another. So we have a measure for modularity. Well, wait a minute, what is it about how efficiently I can get from any particular pair of cities? Like, is it faster for me to go through New York, or do I want to go through the bypass? Well, that's a property called efficiency. Transitivity is a fancy measure for, well, are all the neighborhoods well connected together, right? Am I in a community that works well together? And finally, small world propensity is this idea that our brains balance between the last two things, right? You want to have a pretty efficient network. You'd like to get anywhere pretty quickly. But if you only get places quickly with no deeper processing, that's not really a good processor. So you take both of those last two and you make a ratio, and that's called small world propensity. Okay, so these work in the sense that lots of large studies have linked them broadly to lots of large cognitive outcomes. They say virtually nothing specifically about how anything works. Okay, so keep that in mind for the next part. Here we were just sort of motivating, do we get the variance and can we get it through the measures? Long story short, there are a whole bunch of graphs I'm not going to show you here, but the punchline is basically for any given measure, we took a whole brain assay of that network. We took a left hemisphere assay. We first confirmed that the stroke matched the simulation. That's that pair over here of the orange and the yellow. What we basically want to see is that they match. We want to know the simulation is a similar property to the real strokes. And long story short, with lots of other graphs, what you're seeing here on the y-axis is the WAB-AQ scores, is the overall aphasia quotient, the language performance for these folks. What you're seeing down here is the R-squared value. So this is the proportion of variance explained. So my 20% figure is actually a little bit worse than what we actually found. I'm glad I get to say this. So at about 20% of the variance is just accounted for by looking at the actual strokes. Once we do the simulation trick and populate that estimate with all the other people in the room and what could have happened to them, which gives you a prior on what you'd expect about that stroke overall, well, the variance can creep up. We can get closer to 24 or 25%. So we showed this paper unteased a lot of things about like, well, what way do we image them to do this? Like what measure really accounts for the most weight? Like, is it really about the WAB-AQ or a specific subdomain? I encourage you to read if you get interested in that. But my punchline is sort of, if I just care about the global clinical outcome score, I get a good chunk of variance and maybe the simulation is telling me something I otherwise could not know. And the last detail, you might have noticed this gray distribution in the background. That's a permutation against what if I just shuffled all of the data? What would I have expected? So what you don't see in a lot of imaging studies is just really what would have happened by chance. So we permute that to sort of get a guess. Well, it turns out you could have spuriously gotten somewhere around 10% of the variance anyway, just by coincidence. And so what we really like to see is a continuous measure pushing that out. I'd love for it to be all the way out in the most infrequent outcomes, but we're out here approaching the tail with the simulation. So that's starting to suggest we're pulling out something that maybe is a little structured and real. Okay, so that was encouraging sign one. So then I think about theory again, and what my neuropsychology trainers and folks at Moss were teaching me. I'm like, man, it's starting really to bother me that I'm doing this network thing and I can't get back to what we think we know about the brain. This is a problem, right? It's still a problem, right? We have people doing these studies at scale with measures like this, and it's like, well, wait a second, what about my model for Broca's area and semantics and phonology and whatever process this patient struggles with? Okay. So another line of work has been going on for a while with collaborators at Penn, that's Branch Coslett and Roy Hamilton, where it turns out that when you have some damage to the left inferior frontal gyrus, which is broadly linked to production of language, maybe some semantic control, maybe phonological control and expression, among other outputs we could talk about. Well, what tends to happen is if I'm damaged there, I tend to have this Broca's-like deficit, which again, we're still arguing about the details in which pathways. What also happens is it turns out that in the right homotope, over here in the right inferior frontal gyrus, off in the right pars triangularis, we tend to have a treatment response when I use magnetic stimulation. People tend to get better if I use what we think of as an inhibitory sequence. We try to tone down the region. Classic explanations for this were transcallosal inhibition, people kind of trying to say, well, it sort of works the way the motor system does, which first of all, apparently is questionable to my knowledge, but then you take that to the language circuit and you might expect it to be quite a bit more complex than that. So we were thinking about this problem. I was obsessing about structure and circuits and getting back to this. And so we sort of noticed that something hadn't been asked yet. Well, if you're kind of following what we showed in the first study, kind of very broadly, these big global measures of function, independent from the lesion mass and location, say something about the outcome variance. Okay. So we go back to this case series and we're starting to look at the lesions. And we start to have this idea, well, wait a second. If I blow out something on the one hemisphere, not only does that thing lost, but the structure of the network that's left available by definition has to be different than it was before. Not just at the place I blew out and the connections I lost, but the structure of the roadways that are left. So if you imagine, you can take some of these nodes. I'm not going to get into the details for the sake of time, but we look at this property called boundary controllability, where the first thing we started to think about was, well, lots of people have been showing over and over in the imaging literature, well, it's kind of like there are these special nodes that sort of sit at these boundaries between modules. And you might expect that depending on where you are in the brain, if you knock one out, you're going to disrupt the ability of those big modules to communicate. The additional thing we started to think about was, well, wait a second, if I start to lose that, the whole nature of the modules is different. The nature of the paths is different. The whole topology of the network has changed. We're in a whole new roadmap. You could think about it as like if you wiped out the road infrastructure on the West Coast, right? What's left to use on the East Coast has a completely different network configuration relative to including that. So even though the structure is exactly the same as it was, the demands are completely different. This was the idea we were after. So we started thinking, well, wait a minute. If you start blowing out these things we already know are super important in holding the structure together, where do they go? Are the new highest roles still in the same place? Do they move to some of these other bypasses? And are those shifts related to outcomes that we care about? That was the association we were after. So we took that down more concretely to our friend, the IFG. The conjecture at the time was, is it more than just the direct path? Does this shift in roles tell us something that we didn't already know? So we went in, we found cases, some of whom had more or less direct or indirect damage to the IFG. That's important. We don't only want to look at the lesions that are knocked out at IFG. We were to sort of like sample, well, sometimes we're going to ruin the inputs. Sometimes we're going to hit back closer to Wernicke's area. We have a comprehension disorder, but there's still a referred change in the structure because we lost connections through the arcuate and so on. And what we started asking was, well, if I knock out a particular node and it would have been a strong boundary controller before between these modules, where did it go? Did it go to the ones that were strong before or not? So long story short there, this was with Harrison Stoll, a more senior graduate student with me who was formerly of Moss. They do, they move. And so what tends to happen is we took here on this graph, neurotypical individuals will be here in the sort of bluish color here, persons with aphasia in the sort of yellow-orange color. And we looked at a number of regions, but we lensed in on, well, if I knock it out here, what happens over there? And is that different from somewhere we'd expect to change less? And so we focused in on the pars opercularis down here, pars triangularis here, which is often where people stimulate. And then we just pick a right occipital control region to see if it's selective to these regions. So it turns out there's a little bit of movement, sort of structural recruitment in this role in both the opercularis and the pars triangularis. It's stronger at the triangularis, so the homotope to the region we were interested in. And we started to ask this question, all right, well, that's great. We can kind of show the structure is remapped, which first of all was sort of surprising because it didn't have to work out that it was exactly in that region in a measure like this. Like it could have gone somewhere else. So it's sort of interesting to me that just the idea that homotopes are recruited is a structural network shift somewhere under the hood that we have to look at. That's possible, it seems, from evidence like this. I want to know something about their performance and their outcome. So we started having this tension because in healthy studies about a decade ago, we had already sort of found out that this property was linked to healthy language responses to TMS in the left hemisphere. But there was this weird thing going on. We couldn't kind of separate out in the data. Is that like a semantic effect and it's semantic satiation we're releasing them from? Is that like a semantic control demand that be accelerated or decreased? It was sort of a muddy story at the time. So we'd think about that in the meantime. And in this sample, well, shifts could do a couple of things. Sometimes they could cause traffic jams. And in fact, that's kind of intuitively what I think about even today when like I knock out a roadway, somewhere else there's a traffic jam. But the other thing that happens even when there's a traffic jam is Google Maps is still going to try to find the most efficient path, right? Whatever you have left, there's some possibility that map is navigable, right? And it might be different in each one of us when we have a stroke, right? Your navigable pathways, your roadways available will be different. And maybe the traffic will be a little bit slower, but maybe your outcome's a little better if you have a better pathway. So we dug into this. Another long story short. So we're sort of looking at the shift model and we're asking, well, when the role shifts, we had to measure because we're able to do it with the data. We had to measure about the shift and we started looking first at global outcomes, right? So my intuition was if these measures are telling us something selective about cognitive neuroanatomy, it should be more specific than that. It might still be related. Certainly if I have some sort of production deficit, that might show up in my WAB-AQ. But with the measure, we weren't really finding anything at all. Sort of, well, you can see the shift in the measure and the behavior is not going with that. Then we looked at the Philadelphia naming test was our other candidate, any percent correct. And I'm a little conservative about this. We sort of stopped there. I don't like just like throwing a lot of outcome measures at the wall. So we tried to kind of keep it a global outcome and then something demanding a naming response. We split it by error type, the next level down. And we found this interesting little trend. So when we look at semantic errors, we didn't really find much of this relationship. When we start to actually be specific to phonological errors, now we're seeing a little bit of a slope come off the ground there. And Harrison's pretty sophisticated at permutation testing and other tests himself. So there's some robust covariates in there. And I'm like, I think there's something going on here. It's not the whopping effect, but sort of interesting. We can take that measure and do it. So this led us to speculate because if you follow this relationship, the more of this controllability you have at the right IFG now, the less errors you make. Right? What do people do when they TMS this region? They try to inhibit it. Usually people get better. What the heck's going on there? Okay. So we took that, and in the meantime, Roy Hamilton and I were reading a lot, and we were thinking about like, we've been trying to get this sort of structure, function, cognitive neurology off the ground. We had a couple ill-fated attempts at a couple study sections. And we kind of went back and started looking at some authors who were interested in sort of like executive moderators of these results. What we realized was every time we look at this structural underpinning, the thing we didn't ask was that optimal for the person? Okay, so that was the next and kind of last part of the story here. So we started to think more seriously about, well, what if I wanted to nudge it back on track? And if I postulate that property matters, it has something to do with how well you're going to do in your outcome and in your TMS response. And I'm navigating around, and you're going to hear different versions of this, but we can kind of E-field model the transcranial magnetic field. I'm still alarmed, even when I do this to myself, demonstrating it to undergrads, like I can still pick out the first dorsal interosseous. So there's sort of this strange, pretty ultrafocal, and then like sort of a field splashing around it. So we still tend to use this technique in part for that reason. We can kind of get pretty close to a particular region. We know, because we've done it, we can actually predict these role shifts. We can do even better with that with the simulation than we could before at this point. Does where that moves have something to do with TMS responses? So we had been piloting a very small series of cases where we did a lot of these network partitions prospectively in a small handful of patients over there with Roy Hamilton. And what we started to find was indeed we could move around some of the phonological control measures. We also found that if you actually target the strongest controller, you get more of a semantic response. So what's going on here? The speculation was that in the previous sample, we just kind of fixed the position. We said, well, it was on the left here and now it's on the right there. But what we found in the data was it kind of depends. It's like not every pars triangularis is equal. Not every shift is in exactly the same place. So what if I follow the shift around? What if I say that's the property I need to go through, even though before I would have thought it was just pars triangularis, broadly defined. So what we started to find was we could do this on every single case because we have all of these estimates kind of sitting in the computer in the background, and we can build maps, and we can start to say, well, now that I have damaged this connectome in a way we can draw out with the lesion and then put in that particular connectome, that diffusion scan, we can see where the mass of those rolls goes. We can see where they're recruited. And a part of this well-scored grant that should the federal government resume, we hope we get to actually do. We also had this problem where it's like, how do you cut the network up? So some of you might be familiar with everybody has their favorite network partition. I call this the like, whose network is it anyway phenomena. It's like everybody has their favorite and we keep getting more of them. And this is the function. And here's my argument. And maybe I want to personalize it, but we're all going back to BTT-YEL from like 20 years ago. And it's kind of become this ballooned mess. So rather than take that sort of head on to make a new parcellation or resolve it all, part of what this grant asks is, what if there's a consensus? What if I actually just ask all of the networks what they have in common, and then try to robustly distill from that, kind of like we did from the simulation, the best estimate for all the voxels in the brain. And so that's actually starting to work pretty well. And for the sake of time, I won't get into this, but there's actually quite a bit of structural recruitment in networks that might be familiar to some of you when we do it this way. But the trick is you have to let everybody vote. You have to have everybody have a voice in that network space to get a robust estimate. You can't just kind of pick your favorite and then decide that's the way to do it. You might do something very different if you already know the circuit you're interested in. So this is a sort of a tension for our times and a story for another time. All right, let me wrap up. I don't know how my time is, but this is my last slide, I promise. How do we make it more clinically relevant? I almost want to joke, like, I don't really know, but let me be more helpful than that. There are reasons that this isn't happening, in my opinion now. This is trite, but I think still true. We just aren't doing great at alignment between theory and measures and what they need to do to show up into a clinic. Let me be a little more concrete. I was trained as a clinical neuropsychologist initially. I would sit with the person and do an assessment with them, or even at the time, use a handheld device, and we're interacting, we're talking, we're doing things, right? I'm in the scanner. I mean, like, we're sardines in there. Like, we're trying to like cramp and adapt and like control everything meticulously. This is the same old ecological problem, but not really to me. I still am worried. We're not like 100% sure what to measure at any one time. I'm still worried about that. Some of them are reliable and valid. Some of them are very useful. And I have sort of come around in the sense where I really thought the way to solve this problem was going to be to keep drilling deeper and get all the mechanics. And then I kind of came around where I'm like, just predicting it is pretty great if it works. Let's at least sort of get part of that. So I still think we need to predict and explain, but I think we need to keep talking among clinicians and researchers about what matters to measure. What does it mean to do a good job at that? I can't get around it. I've been trying for 20 years to let the imaging solve my problems. I have to get the behavior. It doesn't go away. And then when we go drift from that, every few years, it's predictable to me at this point. We come back around here like, man, like, we didn't know what behavior to measure. So I just, please lead with the behavior is I beg of all of us, right? The imaging's still not great. It's not perfect. I know there's error in the imaging measures. It has to be there, right? And so that's time. That's probably not for me or most people in the room, some in the room, but like, that's going to keep taking progress. The prediction science is actually like, I don't know when I call it good, but it's gone from bad to like not bad, demonstrably at this point, I could say. So in stroke outcome prediction, including Shreya Parshar over at Penn, whose dissertation committee I sit on, she's very sharp in using these multimodal methods, but sort of took the structure first approach and sort of says, well, let's stack the lesion information, let's take the connectome information, and if we were doing 30 to 40% before, how much do we get out of it? And the committee, myself included, we all had this sort of like tired, wizened perspective where I'm like, it's easy to cheat and you're going to get like spurious inflation and your accuracy is going to look perfect. And then she found another sample, and then she found another out of sample population and kept cross-training the method. Starts kicking this up closer to 80%. So for an imaging researcher, when you do all of those steps, I'm happy because we're doing all of the cross-validating and comparison. As a clinician by training, I'm still worried. We usually don't have great sensitivity specificity, and we usually just need way too much measurement, right? Like, getting somebody to get these high-grade scans, again, is special in the first place. The chances somebody in the real world actually has it are low. So one of the things here, I think, is nobody really solved the n=1 problem. I still kind of use this rhetorical device. The patients here, this is the person I'm treating. So why in the heck are we keep trying over and over to make big population scale research tell us about that? Why wouldn't you start from the person and build from the ground up? And why wouldn't that kind of constrain what you're trying to predict about them? Just a thought we could talk about. Your model-based prediction works on the available data. So again, the advanced models usually need to still work on a clinical routine to actually be helpful, not just like right now, but soon, right? The likelihood that the fMRI gets into every one of your clinical care still seems low to me. We've been trying to do that for 20 years. Sometimes it shows up. Sometimes we're doing imaging guiding stimulation at certain centers, but we don't see a lot of it. And I don't think that's a coincidence. I just don't think we're convinced that we're doing well enough to justify the effort and the cost. I don't think we're there. When we say clinical routine could use more modern methods, I personally now think that onus is squarely on researchers in the sense that we need to do a better job at figuring out what actually matters before we start doing the big grain work. This is, again, just an opinion in my early mid-career, I guess. But like we have to filter the questions down. It's time, it's resources. We need to do better. The network science itself can improve. So I was picking on half of my own field. They're just numbers, right? They're just abstractions built over the network. I've shown you maybe they can be predictively valid. I've said to you maybe from a certain network general theory perspective, they're important. What do they say about the models for cognition and rehab? I am still flummoxed. I am trying, I promise, but I'm sort of like, I kind of like get to a sort of level where like I'm convinced on the prediction. Yeah, generally a network should work like this, but as soon as you go back to that layer about how does phantom limb pain work? How does use capacity discrepancy work? How does TBI work? You need to be more specific than that. You need to acknowledge that there's a brain there with tissue and cells that mediate the whole thing with electrophysiology and neurotransmitters. And I got to say, I read a lot of network studies, and there's sort of progress, but there's never the people that come on and say, let's solve that problem. Let's make the constraint what the brain is, what we know about it, and have our network tools adapt to that. It's sort of infiltrated from the other direction. We got excited, myself included, about networks. Yes, that sounds like it solves the problem, but I think we had to do the other thing first, or maybe at least at the same time. Okay. And so lastly, I am kind of coming around on this idea now that I've seen it work at least once. We have a lot of methods, and I think population voting procedures is a very powerful thing. We've seen that in a lot of sciences. So when I know there are 15 different investigators at reputable places doing good work, why wouldn't I ask them all to have a vote in the process? And so when we start to think about those procedures, let everybody contribute, right? Sometimes it's competitive, ask if one is better than the other. A lot of the time, I think it's actually cooperative. I sound like a bleeding heart up here, but I really am. It's like everybody has something to contribute, but we got to put it together to distill the votes down. All right, I've done talking for today. Happy to take any questions.